Hello, my name is Fayaz Minhas, and I would like to welcome you to what is probably the first virtual digital pathology and AI Congress in Europe. I would like to begin by thanking all the organizers for this Congress and all the attendees who are in audience today. The title of my talk is Whole Slide Images are Graphs. So, what we'll essentially be talking about is another way of representing whole slide images for various types of computational pathology problems. And the talk is aimed both at computer scientists like myself, as well as pathologists who are interested in developing solutions to machine learning based solutions to computational pathology problems. However, before moving on to the more technical details of this talk, I would like to give a brief introduction to myself. So this is me. This is what I look like in case we meet with our masks off sometime after this Congress. And I'm standing right in front of my office at the University of Warwick. It's a pretty beautiful place with plenty of fall colors, as you can see in the background. As part of the Department of Computer Science where I work, I'm engaged in the Tissue Image Analytics Lab. We are a bunch of about 30 or so individuals, all working towards the development of machine learning based solutions, led by Nasir Rajput, who's hiding over here. Uh, the goal of our lab is essentially, as I said earlier, to develop digital pathology solutions. We take in digital pathology data, genomics data, and we want to identify different types of insights and identify different types of signatures using machine learning. I'm also affiliated with the PathLAKE consortium, which stands for Pathology Image Data Lake for Analytics, Knowledge and Education. We are funded with a 50 million pounds grant by the UK government to digitize different NHS sites and to solve their computational pathology problems. So, with that introduction, I'll be switching into the more technical bits of my presentation. So, different types of prediction problems in computational pathology. I think the limit to the different types of prediction problems in computational pathology is only our collective imagination. So there are a very large number of very interesting prediction problems in computational pathology. So essentially, if we can think of them, think of a few examples of them like this. If you're given a whole slide image, like the one that you see on your screen, can we predict the grade of cancer for this patient? Can we identify different types of tumor regions within this image? Whether this person was HER2 positive or not? What is the mutation status of this particular patient? What were the chances of survival for this particular patient? Are there any specific patterns that are associated with patients that have a high chance of survival? And how do different patients react given a drug? How do we identify similar patients, similar images when we give a representative image? How do we find different types of biomarkers? So there's a lot of very interesting problems in computational pathology. And machine learning is a very good framework of modeling or solving these problems. So what we do in machine learning is we collect whole slide image data. We have the target variable of interest for a set of what are called training images. We may also want to get some more detailed annotations for these images. We feed all this data into a machine learning model. So these days, most people work on the development of neural network-based machine learning models. Neural networks are computer algorithms that are based on how our brain identify patterns and how they are connected, how the neurons in our brains are connected to identify these patterns. It's a loose modeling of the same principle done in the computer. So we use all the training data that we have in terms of full slide images. We push it into a neural network, and then we try to predict for a new test case whether the neural network generates a good prediction or not. So that is the performance assessment and validation part. That is whether the neural network is what we call able to generalize to unseen test cases. If that is the case, we can go ahead and deploy it. Otherwise, we can go back into this waterfall and do multiple iterations. We may also be interested in finding different type of correlative and associative patterns that may be associated with our variable of interest. So that is what the typical pipeline for a computational pathology solution looks like. And it's pretty standard in terms of machine learning. Other problems in machine learning are also solved using a similar framework. However, there are a few challenges that are associated with computational pathology only, and I'd like to go over those so that those would form the basis of why we need better ways of representing whole slide images. So if you think of whole slide images, these are pretty large, first of all, and then they are multi-resolutional. So what I've seen pathologists do is that they're able to zoom into the image at different levels to identify different patterns. So depending upon the type of disease they are screening for or the hypothesis they have in their mind, they may zoom to a greater depth within the image and identify those patterns within a given image. Some of the patterns can be pretty obvious at a pretty high level as well. So these images, this modeling, this multi-resolution nature within the machine learning model is key to developing effective solutions. Similarly, as I said earlier, these whole slide images are pretty large, and that presents unique technical challenges as well as machine learning challenges on how these solutions are developed. So typically, neural networks are run on GPUs, which have a limited memory. So fitting a large number of images and then trying to learn over those images may not always be possible. So we need better GPUs with more memory. That's something for the excellent GPU vendors to consider. However, it also presents a set of unique machine learning challenges that I'll talk about in just a minute. Another problem that is associated with whole slide images is that the spatial context in which something occurs matters a lot. So, for example, in colorectal biopsies, if we see a bunch of lymphocytes, those may or may not be malignant depending upon the context of where they occur. So modeling this context of where something is at is also very important in developing effective solutions to computational pathology problems. Typically, it's really easy to get slide level diagnosis from a pathologist for a given whole slide image. However, getting more detailed annotations of different types of tumor regions or for cell level annotations can be quite hard. So the number of annotations that we can expect from a pathologist or the amount of labeling or detailed labeling that we can expect with a whole slide image can be quite small. So we call this the weak supervision problem in machine learning, in which you have a single image and you have the associated label, but we don't know what part of the image is responsible for that particular prediction label. This is another unique aspect of or challenge of computational pathology or of machine learning in computational pathology. So the classical workflow of how people solve these challenges is that they take a multi-gigapixel whole slide image and they chop it into a set of visual fields we call patches. Those some features of those patches are extracted and those are given to a classifier or neural network, which generates essentially a prediction label for each of those patches. The prediction labels of those patches are then aggregated into generating a whole slide level label, which is then associated with a given test case after training. So this is the typical workflow of how things are done. And the aggregation problem is an unsolved problem in machine learning, and there are a bunch of different types of solutions, ranging from taking the most positive patch within a given image to averaging, taking the average of the prediction scores of different patches within an image. There are more technical solutions based on multiple instance learning. There are solutions based on attention mechanisms and long short-term memories, which work to varying degrees of success in different types of problems. However, I relate this to a sort of a analog that you see on your screen, that if we have a bunch of blindfolded people touching different parts of an elephant, each one can come up with something very different, and aggregating their decisions can be quite hard. So you think of each of a neural network when it's given a patch in an independent manner, it may come up with a solution with a prediction that is very different or may not fit with the global context of the whole slide image. So this is the fundamental problem in whole slide image computational pathology based predictions. So I call it the aggregation problem. And so together with this aggregation problem and all the other problems that we discussed earlier, the whole slide images being very large, uh, and then not being able to fit into the GPU memory, having a limited number of annotations, detailed annotations, uh, and all the problems that we talked earlier. These challenges required the development of a specialized type of representation for a given whole slide image. And what we propose is that graphs are a natural way of modeling whole slide images. How is that the case? We'll talk about that in just a minute. But before moving there, let me introduce what graphs are. So if you think of the London Metro, for example, you can think of each station as a node in what we call graph or a network. And then each station has a set of features or that describe that particular station. What's the name of the station? How big is it? How important that station is? How many people go through the station every day, and so on. And then we have edges or a set of edges in the network which connect to different stations. And each of those edges can also have a set of descriptors. How long is the edge? Whether the edge is single directional or multi-directional or so on. So a single graph can be thought of as a set of nodes, each of them having a set of features, and we can have a set of edges, each of them having a set of features. And graphs have been used extensively ever since computer science has been in existence, and even before that. And we think that graphs can be a very natural way of modeling whole slide images as well. So if you think of this whole slide image that you see on your screen, you can take different regions within this whole slide image, and each of them we can treat them that region as a node. And the features of that node can be based upon the local characteristics of that particular region in the whole slide image. And where that node occurs or that region occurs within the whole slide image, we can connect it to other neighboring regions and come up with a graph representation like the one that you see on your screen. Now, the false colors of each node in this graph representation of the whole slide image are based on the local tissue structure at that particular location within the image. And the interconnectivity between different nodes is based upon the spatial arrangement of different nodes or different parts of the tissue that we have captured in the whole slide image. The good thing about this is this can be multi-resolutional. This can, once we have a big enough set of descriptors, we can model different types of details for a given whole slide image, and this makes it very attractive for computational pathology problems. Starting with this hypothesis, we developed an approach called slide graph, in which we solved various computational pathology problems by modeling them as graphs. So what slide graph does essentially is that for a given whole slide image, it builds its graph representation, and then for a given data set, we use specialized types of neural networks called graph convolution neural networks, which are able to handle these graph structures, and then they are able to generate predictions for for each whole slide image, thus overcoming the aggregation problem and solving the other type or overcoming the other challenges that I discussed earlier. So this work was published in CVPR workshop earlier this year, and what it essentially does is that it takes a whole slide image. For each region in the whole slide image, we identify different type of nuclei within that region, and then we cluster those regions based on how similar their nuclear profiles are. So if the same type of nuclei occur in two different regions, then we classify those regions to be similar, and if they are closer together, we cluster them. And then we construct a graph by modeling how different regions are connected to each other. For a given data set with associated training labels, we feed those to train a graph convolution neural network. And once we get a test label, a test image, we do the same process of graph construction and then feed that into the trained graph convolution neural network to generate a prediction label. So that is the brief workflow of how slide graph works. For the first step, as I just discussed, is identifying different types of nuclei. segmenting those, that is identifying where they occur, what is the boundary of those nuclei, and what type of nucleus it is, whether it is an epithelial cell, whether it is an inflammatory cell, whether it is a muscle cell. So we do that using an in-house method that is called HoverNet. It was published back in 2019, and it's a really good method for identifying and segmenting these different type of nuclei within an image. So you're welcome to try it out. Once we have those, we represent the whole slide image in terms of, or different regions in the whole slide image in terms of their nuclear profiles. So that is essentially the count or the density of different regions in the different type of nuclei, different type of cells in a given region, the whole slide image. Once we do that, we do perform clustering. So similar regions within the whole slide image that co-occur, that occur close to each other and have the same nuclear profile, that is, have the same type of cells are clustered together. This is done to reduce the amount of memory that we need in order to process a large number of whole slide images. Once we get these clusters, we interconnect these clusters based on the spatial arrangement of where they occur. So if there's a cluster over here and it is uh, composed of different regions and it is right next to this other cluster, then what we do is that we model these two different clusters as two different nodes in a neural network. Now, the color of the nodes that you see it is dependent upon the type of nuclei that are present within this particular image. So even if you construct uh, a graph for a given whole slide image, this is what we call slide graph, as you can see on your screen. Different regions have different colors, which show different types of cells or the nuclear composition, and this can be a very useful visualization tool as well in order to analyze without going into without zooming too much, identifying different type of patterns in whole slide images. Okay. So here's a bunch of whole slide images represented as slide graphs for various HER2 positive and HER2 negative cases. And as I discussed previously, the false colors that we have assigned to these different regions within these images are based upon their nuclear profiles. And as you can see that different regions in HER2 positive images have different nuclear profiles in comparison to HER2 negative images, which should make their classification possible. However, doing this eyeballing for a large amount of data may not always be feasible, so we use graph neural networks, which are designed to handle such graph data. And what they do is that they take in a graph and then generate a prediction for a given image. And then they can be trained for a given label data set, and we can then generate test predictions for a given whole slide image. So this is the overall workflow. We take a whole slide image, make a graph out of it, and then feed that graph into a neural network, which is trained on a set of images. Once we have this trained neural network, we can then test it on unseen data. We tried SlideGraph, the original version of SlideGraph, on two prediction problems of receptor prediction in breast cancer using a cohort from the TCGA, and we compared it to conventional neural network approaches that do those patches and then try to classify them using classical convolution neural networks. So what we saw is that SlideGraph is able to outperform these classical or conventional methods for both of these problems. Since then, since the publication of our first slide graph paper, we've extended this idea and we've modeled other types of prediction problems, including ER prediction, as well as prediction of different types of colorectal and breast cancer mutations. And what we've seen that is that, I guess, graph-based modeling approach either matches or outperforms published state-of-the-art methods in this domain. So this can be a very useful way of modeling these prediction problems. We've also developed a visualization tool that allows pathologists to visualize a whole slide image in terms of graphs and allows them to zoom into different areas without actually zooming in. So you can see details that are not obvious at first glance. So for example, if there can be some specialized patterns of cells that are occurring, which is different from its neighbors, that is really easy to pick up in the graph representation, but it may require the pathologist to zoom in otherwise. So this can be a very effective way of modeling these graph structures. So with that, I'd like to conclude by putting out this question actually. What do tumors, gene ontology, and risk have in common? What we've seen is that if we model tumors in whole slide images as graphs, then we can use machine learning to identify patterns and solve different type of prediction problems. Now, Go, the classical game of Go, which had recently been solved by an AI called AlphaGo, and Risk, which is a board game in which your objective is to dominate the board, which represents a world. Both of these have an inherent graph structure, and modeling them as graphs has been very successful. So if you're going off on break, I really recommend you either play one or both of these games, Go or Risk, and relate them on how we can analyze tumor whole slide images in the same way. So, for example, if there is a tumor region and there are lymphocytes surrounding it, then this is pretty much similar to what the strategy of Go is, in which our objective is to surround an opponent. So I think I leave you with that, and I really thank you for your patient listening. And if there are any questions, you can contact me via my Twitter handle or send me an email. I would also like to introduce and announce some open CR UK based postdoctoral position with me as part of the PRISM project, which is aimed at the discovery of neoplastic signature of mesothelioma. So if you want to work in this space, please get in touch. Thank you again, and I wish you a very good day ahead. Bye.